All right, let me get my umbrella up. That'll block the sound of the wind into the microphone, perhaps buffer some of the other sounds. Okay, I apologize for all of this, but this is all I have available to me at the time. That's why I'm glad you're reading along with me. Microbiology. Darwin's Black Box. Michael Behe wrote a great book on microbiology and examined it carefully. You see that there had to be a creation of an intelligent designer. It couldn't have evolved. Probabilities are just too lengthy to be even considered. So here we go. Microbiology. Irreducibly complex and demands an intelligent creator. Every door science opens reveals 10 as yet unopened doors. While knowledge of the universe is expanding exponentially, the unknown expands even faster. Like receding images in a hall of mirrors, scientific discoveries overwhelmingly necessitate a power and wisdom, without beginning or end and infinitely beyond human comprehension, which alone could have brought all into existence. Even such a determined proponent of evolution as Richard Dawkins confesses that living things give the appearance of having been designed for a purpose. He admits that the nucleus of every cell, the smallest living unit, of which there are trillions in the human body, contains a digitally coded database larger information content than all 30 volumes of the Encyclopedia Britannica put together. Just the mathematical odds of getting millions of letters lined up in the right order by chance is off the possibility chart. For life, something even more amazing is involved than chance aligning billions of chemical molecules in the right order. Dawkins refers to a digitally coded database. This is recent terminology, never imagined by Darwin. Not only must the DNA molecules be put together correctly, but they must, like letters, express information in a language providing instructions to be followed. Each person at the moment of conception begins as a single cell. How does that cell know what to do to construct a body composed of trillions of individual cells of different kinds and different functions? Most school children know the answer. Imprinted in that original cell are instructions for the construction and operation of the human body, instructions which will be followed unerringly. DNA replicates this blueprint onto every cell produced, and every cell, amazingly, will know which part of those directions it is to follow. Today's school child also knows that DNA has an incredible capacity for storing information. The information contained in DNA, the size of a pinhead, would fill a stack of books 500 times as high as the distance from the Earth to the Moon. It would take tens of thousands of desktop computers to store and process that amount of data. The world's fastest supercomputer is now being completed. It is called Blue Gene, and will perform 1 quadrillion, 1 with 15 zeros after it, calculations per second. It is being built to map the 3 billion chemical letters in the human genome, equally to a 100,000-page run-on sentence of operating instructions for a human being, all put together by chance? Blue Gene's first task will be to figure out how the body makes just one protein molecule. To solve that problem, it will run 24 hours a day, 7 days a week for a full year. Yet the body, following the instructions printed in DNA, creates a protein molecule in a fraction of a second. With the instructions which this computer will take a year to understand arrived at by random processes, all this for just one protein molecule? After all, the computer was pre-programmed. The probability of the required order in a single basic protein molecule arising purely from chance is estimated at 1 chance in 1, followed by 43 zeros. Since thousands of complex protein molecules are required to build a simple cell, probability moves outside of the realm of possibility. The It takes many different kinds of enzymes made of protein to decode, translate the genetic information encoded into DNA, and the enzymes are independently encoded to do this. So it would do no good. So it would do no good for evolution, even if it could, to imprint genetic information on DNA. At the same time, it would have to independently encode the enzymes to translate it. DNA and the enzymes to decode it could not evolve over a period of time. All must be in perfect working order from the start. At the molecular level, evolution is a bad joke. Years ago, the conundrum was, which came first, the chicken or the egg? Now it's which came first, protein or DNA? It takes protein to construct DNA, but it takes DNA to make protein. Obviously, both were created at once. Neither could have evolved. But the lesson of DNA points far beyond the statistical impossibility of it at all somehow falling together through random processes over great time. The 3 billion chemical letters express information in a language which must be read to be usable. A language necessarily involves ideas framed within grammatical rules and can be created and expressed only by intelligence. This moves us beyond statistics and matter into another realm involving issues, and issues cannot be comprehended by tissues. Language expresses thoughts, and thoughts are not physical. They may be articulated in physical form, such as sounds of words, and sentences on a page, or the coded chemical letters in DNA. Obviously, however, the thoughts being conveyed by the language are independent of the material upon which they are expressed. The sentence may be written on paper, wood, sand, a computer chip, or audio tape, but none of these originated the message. It must have an intelligent, non-physical source independent of the physical means of storage or communication. The fact that life is created and functions by language originated from an intelligent, non-physical source forever finishes evolution. There is no way that chemicals could put together intelligent thoughts in language that contains the instructions for constructing and operating even a single cell, much less the trillions of cells in the human body. We are driven by science and logic to admit that life in any form can have its source only in a God who is independent of the material universe. That there cannot be more than one source is provided by the uniformity and universality of the language itself. DNA, of course, does not understand the information encoded into it. It is a mechanism built and programmed by the originator of the encoded language to follow his instructions automatically. And the most complex mechanism built by DNA is the human brain. More advanced than any computer yet built by man, it contains some 100 billion nerve cells connected by 240 miles of nerve fibers involving 100 trillion connections. For all of its complexity, the brain no more originates or understands what it is doing than does DNA. The brain does not originate thoughts. If it did, we would have to do whatever our brains decided. On the contrary, we, the real persons inside, do the thinking and deciding, and our brains take these non-physical thoughts and translate them into physical actions through a connection between the spirit and body that science can't fathom. Wilder Penfield, one of the world's leading neurosurgeons, described the brain as a computer programmed by something independent of itself, the mind. Science cannot escape the fact that man himself, like his creator, must be a non-material being in order to originate the thoughts processed by the brain. But man did not originate thought itself. He did not create himself, nor give himself the capacity to think. We look at one thing in Michael J. Behe's book, Darwin's Black Box. The blood coagulating system is irreducibly complex, ruling out evolution and demanding an intelligent designer. Blood clotting is a very complex, intricately woven system consisting of a score of independent protein parts. The absence of or significant defects in any one of a number of the components causes the system to fail. Blood does not clot at the proper time or at the proper place. Blood clotting is on autopilot, and blood clotting requires extreme precision. When a pressurized blood circulation system is punctured, the clot must form quickly or the animal will bleed to death. If blood congeals at the wrong place or time, though, then the clot may block circulation as it does in heart attacks and strokes. Furthermore, a clot has to stop bleeding all along the length of the cut, sealing it completely. Yet blood clotting must be confined so that the blood clots forms only when and where it is required. Only 2 or 3% of the protein in blood plasma consists of protein complex called fibrinogen. Fibrinogen is only the potential clot material. Almost all of the other proteins involved in blood clotting control the timing and placement of the clot. Fibrinogen is a composite of 6 protein chains containing twin pairs of 3 different proteins. Electron microscopy has shown that fibrinogen is a rod-shaped molecule with 2 round bumps on each end of the rod and a single round bump in the middle. So fibrinogen resembles a set of barbells with an extra set of weights in the middle of the bar. Normally, fibrinogen is dissolved in plasma like salt is dissolved in ocean water. It floats around peacefully minding its own business until a cut or injury causes bleeding. Then another protein called thrombin slices off several small pieces from 2 of the 3 pairs of protein chains in fibrinogen. The trim protein, now called fibrin, has sticky patches exposed on its surface that have been covered by the pieces that were cut off. The sticky patches are precisely complementary to portions of other fibrin molecules. The contemporary shapes allow large numbers of fibrins to aggregate with each other, like tuna cans with indented tops to match another tuna can's rounded shaped narrowed bottom. The fibrin molecules do not aggregate to form a random glob. Neither do fibrins stick randomly. Because of the shape of the fibrin molecule, long threads of form cross over each other, and much as a fisherman's net traps fish, make a pretty protein meshwork that entraps blood cells. This is the initial clot. The meshwork covers a large area with a minimum of protein. If it simply formed a lump, much more protein would be required to clog up an area. Thrombin, which cuts off the pieces from fibrinogen, sets in motion the final step of a controlled process. If the only proteins involved in blood coagulation were thrombin and fibrinogen, the process would be uncontrolled. Thrombin would quickly clip up all the fibrinogen to make fibrin. A massive clot would form throughout the animal's circulatory system, solidifying it, and animals would rapidly perish. To avoid such an unhappy ending, an organism must control the activity of the thrombin. And this leads to a more in-depth look at the cascade system of blood clotting, which enables an organism to coagulate blood effectively when required. The body commonly stores enzymes. Looking at the cascade now. Proteins that catalyze the chemical reaction, like the cleavage of a fibrinogen in an inactive form for later use. The inactive forms are called proenzymes. When a signal is received that a certain enzyme is needed, the corresponding proenzyme is activated to give the mature enzyme. As with the conversion of fibrinogen, the fibrin proenzymes are often activated by cutting off a piece of the proenzyme that is blocking a critical area. This strategy is commonly used with digestive enzymes. Large quantities can be stored as inactive proenzymes, then quickly activated when the next good meal comes along. Thrombin initially exists as the inactive form, prothrombin. Because it is inactive, prothrombin can't cleave fibrinogen, and the animal is saved from death by massive inappropriate clotting. Still, the dilemma of control remains. If fibrinogen and prothrombin were the only proteins in the blood clotting pathway, again our animal would be in bad shape. When the animal was cut, prothrombin would just cut helplessly by the fibrinogen as the animal bled to death. Because prothrombin cannot cleave fibrinogen or to fibrin, something is needed to activate prothrombin. Perhaps the reader can see why the blood clotting system is called a cascade, a system where one component activates another component, which activates a third component, and so on. More on this later.